we are interested in the lab to understand fundamental principles that lead to the self-organization in biology. One thing that we are looking at is early development of living organisms in general. And one thing that I found totally fascinating is how if you look at the development of any type of animal, you start with a single cell, and then the cell divides, and you reach an organism that has 1,000 cells, even tens of thousands of cells. And from this group of cells that all have the same genome, the same code, they will afterwards shape an organism and then form different organs. So some part will become eyes, some will become your liver, your stomach, or your heart. And we're interested in understanding how those cells basically succeed to generate shapes. That's what we call morphogenesis. In this particular work, we looked at a specific process of reshaping that occurs during the early development in Drosophila embryo. It's called dorsoclosure. So it's a process that has been looked during the last 10-15 years because it's exactly the same than when you cut yourself and the skin would repair. And that's the mechanisms of this process are exactly the same. So it's like you could imagine like the skin of the embryo that basically would repair itself and would close in order to seal the organism, the embryonic flag. And so what we found is that surprisingly in that particular tissues, forces were generated by a dying tissue that covered the gap. So in that particular case, we found that the cells that were covering the gap were all taking the decision to die altogether, and by dying, they were reducing their size, reducing their volume, generating forces to pull and to seal the skin. This discovery has this to be beautiful in the fact that this is a very general process that is associated to cell death in general. And many processes in development, like for example brain development, involve massive cell death as well. So we could imagine that all the sculpting, the fact that cells die, generate forces to sculpt your brain, or could generate forces to sculpt your brain. And this is also the case where your wound is closing. What happened in the case of your wound is that cells migrate there, then they attach to the extracellular matrix, pull on the tissue, and then activate cell death at this moment. And so we could perfectly imagine they also shrink and then generate forces. So that's a very simple, generic mechanism by which cells could change their volume to basically pull or push tissues. So that's what we found in this particular case. One thing that I find beautiful in biology is the properties of tissues and of repairing themselves. And basically that's why we are interested in looking at this particular process, is because we are interested in understanding at the mechanism that would lead to this perfect repair. And it actually, if you think about it, not only in terms of biology, but in terms of more general aspect, it actually goes much way beyond biology, because you would love to have objects. Like for example, I think about we have in a smartphone technology now, like screen that would repair themselves without not any scar when you break them. So that's like one of the questions that I'm going, even if it's a lot of speculation, is whether the principle that we would find in terms of these self-repair properties of the biological system could be applied to design new materials that we could then use in our daily life, or for a specific experiment that would be to send objects in space, for example, and that basically would repair themselves without our control.